Hello everyone, my name is Alper Tunga Özdemir. I work as a medical biochemistry specialist at Merkez Efendi State Hospital in Turkey. First of all, I thank the organizing committee of Beyond Sciences Initiative for giving me this opportunity. In this presentation, I'm going to tell you about our study on mesenchyme stem cells and costimulatory ligand expressions. Let's start with information about mesenchyme stem cells. What are the mesenchyme stem cells? Mesenchyme stem cells can isolate from many tissues like as umbilical cord, dental pulp, adipose tissue, or bone marrow. These cells are unique because they are multipotent and can differentiate to several cell types such as adipocytes, chondrocytes, or osteocytes, etc. However, the most prominent features of mesenchyme stem cells are their immunomodulating abilities. Mesenchyme stem cells can suppress almost all immune cells in non-selective and non-specific manner. Immunomodulatory effects of mesenchyme stem cells on the adaptive immune cells such as T helper and B cells occur by specific secretome that consist of prostaglandin E2, IDO, hepatocyte growth factor, soluble HLA-G, interleukin 10, and transformed growth factor beta. In this way, pro-inflammatory T and B lymphocytes are suppressed, but regulatory T lymphocytes are stimulated. Numerous clinical studies are conducted with mesenchyme stem cells due to their immunomodulatory abilities. Although promising results have been obtained in the clinical studies, there is no mesenchyme stem cell product has passed phase 3 stage to date. And benefit ratios of patients are variable. The effect is mostly temporary. Why are the effects of mesenchyme stem cells so variable? There could be many different reasons for this. Variables such as the type of tissue in which mesenchyme stem cells are isolated, age and gender of the donors, administration routes, and production techniques may affect the results. However, in this presentation, we will evaluate these in terms of mesenchyme stem cells and adaptive cell interactions. I want to start with some information about the T-cell activation and peripheral tolerance. Antigen presentation is required to activate T cells or induce tolerance. In the first step, MHC molecule complexed with the antigen on the surface of antigen-presenting cells, or APCs, binds to specific T cell receptor or TCRs on the surface of T cells. This interaction is called as signal 1. In addition to signal 1, a second signal is required. Signal 2 is triggered by the interaction of the costimulatory ligand on the surface of antigen-presenting cells and its receptor on the T cell surface. If signal 2 is blocked somehow, anergy occurs against the presented antigen. This is a common form of peripheral tolerance to self-antigens. In the last stage, cytokines secreted or present in the environment determine the type of T cell response, like Th1 or T helper 1, Th2, Th17, or regulatory T cells. This process is called signal 3. Numerous costimulatory pathways that cause inhibition or activation of T cells have been identified. CD1886, inducible costimulatory ligand, or ICOS ligand, 4-1BB ligand, OX40 ligand are prominent activatory ligands. Programmed that ligand 1 and 2 are prominent inhibitor ligands. What about mesenchyme stem cells? In our study, we performed in vitro experiments to answer this question. For this, we investigate change in the expression of costimulatory ligands of mesenchymal stem cells preconditioned with inflammatory cytokines interferon gamma, interleukin-4, and interleukin-10. For comparison with professional antigen-presenting cells, THP macrophages preconditioned under the same condition were evaluated as controls. After the culture period, we analyzed the cell surface alteration of CD80, 86, CD140, 134 ligand or 41BBL, OX40 ligand, PDL1 equals ligand by flow cytometry method, and also HLA ABC and HLA DRDP-DQ. We found that the positive cell frequencies of THP1 and mesenchyme stem cells for HLA1 were similar, and they were not significantly affected by cytokine stimulation. However, HLA2 expressions of both cells increased significantly with cytokine stimulation. On the other hand, changes in HLA2 expressions of MSCs were significantly lower compared to THP1 cells. These data suggested that mesenchyme stem cells do not express HLA2, and even with cytokine stimuli, they may not reach the levels of antigen-presenting cells. Similar conditions were found the expressions of CD80, 86, CD137 ligand, and CD275. The expressions of CD80, 86, and CD275 of THP1 cells increased significantly with stimulation of interferon gamma and interleukin-4, but decreased significantly with interleukin-10. However, we observed that a very low proportion of mesenchyme stem cells express these molecules, and their levels are incomparably low with THP1 cells. The expressions of CD274 and CD252 of THP1 cells increased significantly with stimulation of interferon gamma and interleukin-4, but decreased significantly with IL-10. The expression of these molecules in mesenchyme stem cells were significantly lower than THP1 cells, but at higher levels compared to other costimulatory ligands. In addition to flow cytometry analysis, we made qPCR analysis. The CT values of the CD80, 86, and HLA2 molecules of mesenchyme stem cells were excluded because they were above 35. Although GAP-DH CT values of THP1 and mesenchyme stem cells were similar, there were significant differences between CT values of HLA1, CD275, 274, 252, and 137 ligand. These data suggest that the expressions of costimulatory ligand of mesenchyme stem cells remain significantly lower compared to an antigen-presenting cell, and this may be insufficient to generate an effective signal too. We found that gene expression in both cells increased with interferon gamma. However, we found that interleukin-10 stimulation in THP1 cells and interleukin-4 stimulation in mesenchyme stem cells led to a significant decrease in most of key gene expressions. Mesenchymal stem cells could provide effective immunosuppression through the powerful molecules they secreted, that is, mesenchyme stem cells are strong signal to producer cells. However, this effect is unfortunately limited by the presence of mesenchyme stem cells. As we found in our study results, mesenchyme stem cells and antigen-presenting cells or THP1 cells had similar levels of HLA1 expression. In this way, they can have the potential to induce energy on CD8 T cells that interact with HLA-1. On the other hand, HLA-2 expressions of mesenchyme stem cells are almost absent, and cytokine stimuli do not provide adequate HLA-2 expression. Therefore, they cannot interact with CD4 T cells, which are key cells of adaptive immunity. Thus, when the suppressor cytokines secreted by mesenchyme stem cells are lost, the effects of autoreactive T cells may be restored. To sum up, mesenchyme stem cells are potentially energy-inducing cells due to lack of costimulatory ligand expressions in addition to suppressive cytokine secretions. However, since they express only HLA-1, their effects will be limited to CD8 T cells energy. In addition, since the antigen presented belongs to molecules expressed by mesenchyme stem cells, the energy that will occur will be directed to mesenchyme stem cells itself. Furthermore, they may not be able to induce energy for CD4 T cells due to lack of HLA-2 expression. By transferring the patient's HLA-2 gene to mesenchyme stem cells and loading autoimmune disease-related antigens, the energy induction in CD4 T cells may be achieved. In this way, the effect created by mesenchyme stem cells might result in a persistent peripheral tolerance. This is my final slide. Thank you for patience and attention. I would like to thank the other researchers who participated in the study, especially Professor Dr. Cengiz Kırmaz and Dr. Rabia Bilge Özdemir, and Manisa Celal Bayar University for supporting our study. Thank you very much for